Let's move on to our next speaker, who is Dr. Stephen Burley. Stephen is an expert in data science and bioinformatics, structural biology, and structure-guided drug discovery for oncology. He's the director of the RCSB Protein Data Bank, and also within Rutgers, the State University of New Jersey. He serves as University Professor and Henry Rutgers Chair, also founding director of the Institute for Quantitative Biomedicine and Cancer Pharmacology Research Program co-leader within the Rutgers Cancer Institute of New Jersey. He has published extensively in data science and bioinformatics, artificial intelligence, machine learning, structural biology, and clinical oncology. Today, he will talk to us about Protein Data Bank, from 2 epidemics to the global pandemic to mRNA vaccines and Paxlovid. Stephen, welcome. We're excited to have you here. The floor is yours.
Thank you much. Thank you very much, Serene. Can I start my video? Am I allowed to start my video?
Yes, please do.
Let me just share my screen. Just take me a moment, sorry.
No worries, love the background, Stephen.
And thank you. Sure. You should now see my first PowerPoint slide, do you?
We do. Thank you so
Wonderful.
much.
Thank you very much. Very grateful for your kind introduction and for the honor of presenting in the, let me just do one more thing, sorry, for the honor of presenting in this NIH NCATS in Silico Drug Discovery Workshop. Today, I'm going to present real-world examples of the importance of data preservation and fair sharing. In this talk entitled Protein Data Bank, from 2 Epidemics to the Global Pandemic to mRNA Vaccines and Paxlovid. My plan is to tell you about the critical role that structural biologists and the Protein Data Bank, the PDB, together played in fighting the COVID-19 pandemic. The punchline of my talk was put succinctly by Dr. Anthony Fauci, requiring no introduction to this audience. In the New York Times Magazine in 2023, he cited the importance of 3D biostructure information and said, "Show me a person who's vaccinated, got infected, took Paxlovid and died. I can't find anybody." I'll explain why Fauci made these statements and put the global efforts of structural biologists and the PDB into context. I'll cover the history of the Protein Data Bank and the Worldwide Protein Data Bank Partnership, the RCSB Protein Data Bank, my organization, the U.S. Data Center for the WWPDB, our one-stop shop for public 3D biostructure data, rcsb.org, and then spend the bulk of my time talking about how structural biologists and the PDB played decisive roles in the fight against SARS-CoV-2 and taming of the COVID-19 pandemic. I will close with a sobering postscript. The PDB was established as the first open access digital data resource in all of biology just over 53 years ago. It was founded in 1971 with just 7 X-ray crystal structures of proteins and has been continuously funded by the US government ever since. 4 of the early structures motivating protein crystallographers to establish the PDB are illustrated in the right panel. In biology, function follows form. This variation on the famous phrase coined by the Boston architect Louis B. Sullivan explains succinctly that the function of a biological macromolecule is determined by its form, its shape, its 3D atomic level structure. Since its inception, the archive has grown more than 30,000-fold to become the single global resource for experimentally determined atomic level 3D biostructure information. At present, the PDB houses more than 225,000 structures of proteins, nucleic acids, viruses, and macromolecular machines. Recognizing the global importance of the PDB, it has been managed jointly since 2003 by the Worldwide Protein Data Bank Partnership, including data centers in the US, Europe, Japan, and the People's Republic of China, and 2 specialist data repositories for 3D electron microscopy and NMR spectroscopy. The RCSB Protein Data Bank serves as a U.S. data center for the Worldwide Protein Data Bank Partnership and as the designated WWPDB archive keeper for the Protein Data Bank Core Archive. It is headquartered at Rutgers University in New Jersey with 3 additional performance sites at UC San Diego and UC San Francisco. We operate 5 interlocking services that together convert global data into global knowledge for the public good. Once 3D biostructure data are archived in the PDB, our rcsb.org research-focused web portal serves as a one-stop shop for open access to more than 225,000 experimentally determined PDB structures, alongside more than 1 million computed structure models coming from AlphaFold DB and the Model Archive. We deliver public domain information to many millions of basic and applied science researchers, their trainees and educators, and their students around the world at no cost with no limitations on usage. For the avoidance of doubt, the provenance and reliability of both types of 3D biostructure data, experimentally determined and computationally predicted, are clearly identified on our website. PDB data are essential for responding to emerging viruses. Since the early 2000s, the world has faced down 3 major outbreaks of coronaviral infections that jump the species barrier to humans. First detected in 2002, the SARS coronavirus infected about 8,500 individuals worldwide, killing just over 800 people between 2002 and 2004. Hereafter, I will refer to SARS-CoV as SARS-CoV-1 to avoid confusion. The second coronavirus epidemic caused by the Middle East Respiratory Syndrome or MERS coronavirus struck first in 2012. To date, it has killed more than 900 individuals and remains a public health threat in the Middle East and parts of Asia, where it is endemic in camel populations. Despite these 2 warning signs, very few countries prepared for the possibility of a much more serious coronavirus epidemic. Fortunately for the global community, structural biologists and the PDB laid the groundwork for a successful response to the inevitable 3rd coronavirus wave. Effective mRNA vaccines were designed and antiviral agents were discovered and developed with the benefit of open access to PDB structures of SARS-CoV-1, MERS-CoV, and SARS-CoV-2 proteins. SARS-CoV-2, the unseen enemy, is shown on the right within a respiratory droplet in a haunting painting by our artist-in-residence David Goodsell, who has authored the RCSB PDB Molecule of the Month series for more than 20 years. The organization of the SARS-CoV-2 genome is depicted schematically in this slide. All coronavirus genomes are very long, single-stranded, positive sense, 5' capped, 3' polyadenylated messenger RNAs ready for translation by host cell ribosomes. Most of the non-structural viral proteins are expressed within a pair of related polyproteins. Individual non-structural proteins are excised from the polyprotein by 2 SARS-CoV-2 proteases. Both are non-structural proteins themselves and are part of the polyproteins. The papain-like proteinase cuts at 3 sites denoted with dark blue down arrows. The even more important main protease cuts at 10 sites denoted with light blue inverted triangles. Although we did not know it at the time, pathogen preparedness for SARS-CoV-2 began in the 2000s with work on SARS-CoV-1. Quite rightly, the first PDB structure of a SARS-CoV-1 protein was that of NSP5, the main protease, or MPRO, PDB ID 1 Quebec 2 Whiskey using the NATO alphabet. I say rightly because without the main protease, the viral polyprotein cannot be cleaved to release most of the non-structural proteins, thereby stopping an infection in its tracks. This structure was determined in 2003 by my company, SGX Pharmaceuticals Incorporated, where I led R&D as the chief scientific officer. It has the distinction of being the only PDB structure with its primary publication appearing in the New York Times. Immediately upon deposition to the PDB, my company released the structure to the public so that work on viral countermeasures could begin without delay. To date, more than 90 APO and co-crystal structures of this SARS-CoV-1 protease have been contributed to the archive. The first PDB structure of the entire extracellular portion of the homotrimeric SARS-CoV-1 spike protein, ID 5 Whiskey Romeo Golf, was determined using single-particle cryo-electron microscopy and only released in 2017. To date, more than 70 SARS-CoV-1 spike protein-related structures have been contributed to the archive, including pre- and post-fusion conformations and complexes with its cellular receptor, angiotensin-converting enzyme 2, or ACE2, and various antibody FAB fragments. The first PDB structure of a MERS-CoV protein released in 2013 was that of the ACE2 binding domain of the spike protein shown here in the right panel in a ribbon representation. To date, for MERS-CoV, more than 70 spike protein-related structures have been contributed to the archive, alongside more than 40 M-Pro structures and more than 10 PL-Pro structures. I'll return to the SARS-CoV-1 and MERS-CoV spike protein structures later in my talk because they played critical roles in guiding SARS-CoV-2 vaccine design. Since the very first SARS-CoV-2 protein structures were deposited to the PDB in early 2020, the RCSB Protein Data Bank has been hard at work responding to the ongoing pandemic. Together with our worldwide Protein Data Bank partners, we prioritize complete deposition, rigorous validation, and expert biocuration of COVID-19 related protein structures and strongly encouraged immediate pre-publication release of these data. I'm pleased to report that most structural biologists did release their COVID-19 PDB structures to the public before they were reported in scientific journal articles. We created a coronavirus resources page on our rcsb.org web portal, which is updated weekly with each release cycle, adding approximately 300 new structures of any stripe to the archive every Wednesday. We also developed education and outreach resources, such as this animated hand-washing video, viewed more than 500,000 times, and a series of RCSB PDB Molecule of the Month articles authored by David Goodsell, featuring the proteases, the RNA-dependent RNA polymerase, and spike proteins. The right panel of this slide summarizes our now nearly complete atomic-level 3D knowledge of SARS-CoV-2 protein structures embodied in more than 4,500 PDB structures released since early 2020. That impressive number represents nearly 2% of the entire PDB archive. The left panel shows an artist's rendition of the lipid bilayer of SARS-CoV-2 fusing with the plasma membrane of a soon-to-be infected cell in another haunting painting from David Goodsell. This viral entry process is blocked by the immune response to mRNA vaccines against SARS-CoV-2 that I'll come on to soon. The very first PDB structure of a SARS-CoV-2 spike protein, ID 6 Victor Sierra Bravo, was released in February 2020. Today, there are about 1,900 SARS-CoV-2 spike protein-related structures in the PDB. Like earlier coronavirus spike protein structures, they provide important insights into receptor binding, fusion with the viral lipid bilayer, with the plasma membrane, and vaccine and antibody design. The very first mRNA vaccine designs were generated in January 2020 based on PDB structures of the SARS-CoV-1 and MERS-CoV spike proteins, which are very similar in amino acid sequence and 3D structure to their SARS-CoV-2 counterpart. When designing Moderna's inaugural mRNA-1273 vaccine encoding the SARS-CoV-2 spike protein, researchers at NIAID and the for-profit company reported reliance on PDB structures of double proline modified forms of the SARS-CoV-1 and MERS-CoV spike proteins. This engineered double proline mutation stabilizes both spike proteins in a highly immunogenic pre-fusion conformation. I believe the vaccine designers at BioNTech used the same information because their inaugural mRNA vaccine also encoded a SARS-CoV-2 spike protein bearing the identical double proline change. The rest of the story is well known to you. Two essentially identical, highly effective mRNA vaccines against COVID-19 were brought to the market in record time. To date, these vaccines and their successors have been administered to more than 5.5 billion individuals worldwide with impressive results. The vaccines designed with the benefit of open access to PDB structures are credited with saving tens of millions of lives globally and preventing severe forms of infection in hundreds of millions, if not more than 1 billion individuals worldwide. You've got to love the Protein Data Bank. I'll now turn from the spike protein and vaccine design to discovery of small molecule anti-SARS-CoV-2 drugs. Early in the pandemic, drug repurposing proved to be a successful strategy for targeting the SARS-CoV-2 RNA-dependent RNA polymerase shown in the right panel, ID 6 Yankee Yankee Tango. The viral holoenzyme consists of 4 non-structural proteins, including the smaller NSP7, the larger NSP12, the RNA polymerase subunit, and 2 copies of NSP8, the molecular chopsticks holding the yellow-orange RNA duplex. Gilead's remdesivir was originally developed for treatment of hepatitis C virus infections. It received emergency use authorization for SARS-CoV-2 from US FDA in 2020. The right panel shows PDB-ID 7 Bravo Victor 2 with remdesivir incorporated into the RNA product of the holoenzyme. This structure explains the mechanism of action of the repurposed drug at the atomic level in 3D. Similarly, Merck's molnupiravir was originally discovered at Emory University during work targeting Venezuelan equine encephalitis virus, EEE as it's known, now a public health threat in the United States. It received emergency use authorization for SARS-CoV-2 from the US FDA in 2021. The right panel shows PDB-ID 7 Oscar Zulu uniform with molnupiravir incorporated into the RNA product of the holoenzyme. Like remdesivir, this structure shows the mechanism of action of the repurposed drug at the atomic level in 3D. Moving on, I'll now describe structure-guided drug discovery for SARS-CoV-2 main protease, the Achilles heel of the virus. It is, the target of Pfizer's highly effective, orally bioavailable small molecule drug Paxlovid, which is a fixed-dose combination of nirmatrelvir, the active ingredient, and ritonavir. The PDB currently houses more than 1,500 crystal structures of SARS-CoV-2 Mpro, the first of which was ID 6 Lima Uniform 7, contributed in January 2020 by colleagues based in Shanghai, approximately 2 weeks after the viral genome sequence became publicly available. The nirmatrelvir origin story begins in the early 2000s with discovery and development of PF-00835231, shown here in the right panel inhibiting SARS-CoV-1 Mpro. This would-be Pfizer drug was intended for use as an injectable, in-hospital antiviral agent for acutely ill SARS-CoV-1 infected patients. It was the product of a structure-guided drug discovery campaign initiated in 2003 that was facilitated by open access to the SGX structure of SARS-CoV-1 Mpro, PDB-ID 1 Quebec 2 Whiskey, that I introduced earlier. Regrettably, but understandably, Pfizer halted the project in the mid-2000s because SARS-CoV-1 had disappeared, and the company correctly judged that there would be no commercial market for such a drug. In early 2020, as the pandemic hit, Pfizer quickly reactivated the SARS-CoV-1 project with the goal of discovering and developing an orally bioavailable antiviral agent for outpatient use, facilitated by open access to PDB-ID 6 Lima Uniform 7, the first SARS-CoV-2 Mpro structure I described earlier. The result was nirmatrelvir, shown here in the right panel, inhibiting SARS-CoV-2 Mpro. You've got to love the Protein Data Bank. Both nirmatrelvir and PF-00835231, shown on the previous slide, are covalently acting inhibitors of the protease, binding to the invariant active site cysteine residue visible within the red circle. Upon encountering the drug, the enzyme catalyzes a single turnover reaction, forming the inactive covalent adduct displayed in the right panel. thereby preventing process of the viral polyprotein and stopping the infection in its tracks. It is structurally similar to Pfizer's first-generation SARS-CoV-1 Mpro inhibitor, with key chemical differences that allow it to be administered as a pill on an outpatient basis to newly infected individuals. Nirmatrelvir does, however, have one major drawback. It is rapidly metabolized by the cytochrome P450 isozyme CYP3A4. Co-administration with ritonavir, a potent CYP3A4 inhibitor, prolongs the half-life of nirmatrelvir by preventing degradation as it passes through the liver. Paxlovid, a fixed-dose combination of the two drugs, received emergency use authorization from US FDA in 2021, less than 2 years after public release of the viral genome sequence. The speed with which Pfizer was able to move was unprecedented. At the end of my talk, I'll come back to the fact that Pfizer reported that nirmatrelvir is also active against the main protease of both SARS-CoV-1 and MERS-CoV. This important finding leads me to a sobering postscript. The PDB archive houses more than 100 apo and co-crystal structures of the human cytochrome P450 isozyme CYP3A4. The right panel shows a co-crystal structure of CYP3A4 inhibited by ritonavir. The drug was originally discovered and developed by Abbott, now AbbVie, as an HIV protease inhibitor. This structure explains in 3D at the atomic level why ritonavir is also used for prolonging the half-lives of other viral protease inhibitors susceptible to CYP3A4 degradation. The fact that nirmatrelvir inhibits the main proteases of SARS-CoV-2, SARS-CoV-1, and MERS-CoV is not surprising. In 2021, 4 summer undergraduates working at Rutgers with me and Professor Sagar Kare, our goal was to understand Mpro amino acid sequence, 3D structure, active site, and peptide substrate cut site diversity across all known coronaviruses. This work's not been published, so I'll summarize our results in some detail. All coronavirus main proteases fall into one of 4 genera, as shown in this cladogram. SARS-CoV-1, MERS-CoV, and SARS-CoV-2 all fall within the beta coronavirus genus, denoted in light blue. Please take a moment to note how close SARS-CoV-1 and CoV-2 are to one another within the cladogram. This image shown in the right panel explains how the Mpro active site, revealed by PDB ID 6 Lima uniform 7, can be partitioned into 6 pockets or subsites designated S1' to S5. The invariant active site cysteine visible within the red circle is located between S1' and S1. Like full-length Mpro amino acid sequences, all coronavirus main protease active sites fall into one of 4 distinct genera, as shown in this cladogram, based on clustering of the amino acid residues that line the enzyme active site. Again, CoV-1, MERS-CoV, and CoV-2 all fall within the beta coronavirus genus, shown in light blue. You can see from the detail of this cladogram that the amino acids lining their Mpro active sites are more similar to one another than are the full-length amino acid sequences. Conservation of the amino acid residues lining the main protease active sites of all known coronaviruses is nothing short of remarkable. Each column in the right panel shows the frequency of each of the 20 possible amino acids occurring at each residue lining each enzyme active site. Dark blue identifies amino acids that do not occur at that position, and there's a lot of dark blue in this plot, reflecting the fact that Mpro active sites are highly conserved across all known coronaviruses. For example, SARS-CoV-1 and SARS-CoV-2 active sites are made up of exactly the same amino acid residues occurring in exactly the same positions. There are 12 amino acid residues that are invariant across all known coronaviruses. Based on computed structure modeling using all known coronavirus Mpro structures housed in the PDB, you can see in the right panel that the Mpro polyprotein cup site substrates modeled into PDB ID 6 Lima uniform 7 consist of 6 amino acid residues that I've designated P1' to P5. Protease catalyzed cleavage occurs between P1 and P1', in each of the 10 cut sites distributed through the length of the polyprotein. The invariant active site cysteine is visible within the red circle, where it's positioned to break the peptide bond between P1 and P1 prime.
Just a quick warning, a few more minutes left to wrap up.
I'll be done almost immediately. Main protease polyprotein cut site amino acid sequences are also highly conserved. Each column in the right panel shows the frequency of each of the 20 possible amino acids occurring at P5 through P1 prime. To jump to the bottom, there's double evolutionary selection pressure at work on both the 3D structures of coronavirus MPRO active sites and the amino acid sequences of the 10 MPRO cut sites found in the coronavirus polyprotein. During viral evolution, catalytic activity of the enzyme has to be preserved, and each of the 10 polyprotein cut site amino acids cannot change significantly. We therefore predict that nirmatrelvir will be effective against many of the known coronaviruses, not just CoV-1, MERS-CoV, and CoV-2 as shown by Pfizer. Going beyond their work, we're currently testing these in silico predictions in representative main proteases from a diverse array of coronaviruses. Understanding constraints on the evolution of coronavirus main protease active site structures in 3D and the polyprotein cut site amino acid sequences also tells us that the free market for emergency pharmaceuticals failed us in this case. The free market is defined as an inefficient distribution of goods and services within the economy. Such phenomena occur when individual incentives for rational economic behavior do not lead to a rational outcome for the outcome of society. Simply put, we cannot always rely on capitalism to get the right outcome when it comes to the public good. For better pandemic preparedness benefiting the entire world, Pfizer could have been incentivized with public money in the 2000s to continue working on discovery of broad-spectrum MPRO inhibitors. With the benefit of hindsight, we know that investment of a few $100 million in a SARS-CoV-1 main protease inhibitor could have saved millions of lives worldwide before the mRNA vaccines became broadly available and prevented economic losses to the tune of trillions of US dollars. When the MERS-CoV epidemic struck, there should have been an informed discussion regarding the opportunity and desirability of public investment in pharmacologic countermeasures targeting new coronaviruses. Experts knew that it was no longer a matter of if, but simply a matter of when the next coronavirus epidemic would strike, with the attendant risk of becoming a global pandemic. Thanks to the structural biology community and the open access PDB, experts also knew that the active sites of CoV-1 and MERS-CoV were nearly identical in 3D structure, and that their polyprotein cut site amino acid sequences were highly similar. The free market failed us, but structural biologists did not let us down, and the Protein Data Bank stored the information. So we have Pfizer making sizable investments in structure-guided drug discovery and development of SARS-CoV-1 MPRO inhibitor, which set the stage for very rapid discovery and development and emergency use authorization of Paxlovid. Similarly, the federal government, Moderna, and Pfizer all invested heavily in developing mRNA vaccine technologies. One of the few silver linings from the pandemic is the fact that vaccine discovery and development has been revolutionized. Over the next few years, we can expect to see new mRNA vaccines come onto the market, offering fresh hope to millions of patients and their families, particularly in the area of cancer. I'm also hopeful that there'll be a more concerted and a more effective approach to pandemic preparedness worldwide. To recap, I've explained how open access to research data generated with public and private funds, particularly thousands of PDB structures, enabled basic and applied researchers to combat the COVID-19 pandemic and save tens of millions of lives. Please join me and Dr. Fauci in loving the Protein Data Bank. I thank you for your attention, and I thank all the people depicted in this slide who work at RCSB PDB and our Worldwide Protein Data Bank colleagues who made all this possible. Our funders, NSF, NIH, and DOE are listed here, and as are our hosts at Rutgers, UCSD, and UCSF. Thank you very much for your attention.
Thank you so much for a wonderful talk, Stephen. If you can turn your camera on so we can take a question or two.
Sure.
Perfect. So let's take one or two questions. We can't take more questions because we'll bleed into a break, but here's one. Is there any publication that details the protocols, processes of PDB curation?
There is a publication, Young et al. It appeared in the journal Biocuration that documents all of that. We also have on our worldwideproteindatabank.org website a detailed explanation of both the validation and curation processes that are undertaken when every new structure comes into the protein data bank.
Fantastic.
And I should say that these protocols for validation and biocuration are globally uniform among our colleagues in Japan, the People's Republic of China, Europe, and the US.
Fantastic. Thank you. Next question, how to determine active sites of a protein with no reference structures or native initial ligands? Is there any software or website that can help with this?
So Janet Thornton and her team did an analysis of enzyme active sites many years ago. I don't recall where it was published. And I think the thrust of their results was that the deepest cleft on the surface of the protein is likely to be the enzyme active site when you're dealing with a catalytic entity.
Fantastic, thank you. Next question, we'll take one more. Thank you. So RCSB offers 3D structures in PDB file format, along with other formats. Many researchers favor the PDB format, but it does have its limitations. Are there any plans to modify the PDB format or to introduce a new format that can include more detailed information about proteins in a user-friendly way?
Excellent question. That's already happened. The archival format for the PDB is the PDBX MMSIF data standard. It's closely related to its derivative, the model SIF data standard that's used by AlphaFold DB and by the Model Archive, ITASR, etc. The advantage of the PDBX MMSIF data standard is that it's both machine and human readable. It's infinitely extensible in terms of structure, size, and complexity, and also in terms of the addition of new data standards to support new types of experiment and things like post-translational modifications, etc. So I know that the 80-column Hollerith card, punch card format of the original PDB format is very easy for people to use because they're familiar with it. But I think that in the long run, we all have to move to the PDBX MMSIF data standard if we want the PDB to be useful going forward. Many of the structures that we're now archiving in the PDB cannot be depicted in the old PDB legacy format. So those structured data files are no longer available in legacy PDB format. They're only available in the PDBX MMSIF data standard. So I urge all the software developers on the line and those people who are consuming PDB data to go with the flow, and you'll find that the PDBX MM data SIF standard is easy to learn, easy to read, and easy to work with from a software engineering standpoint.
Fantastic. Thank you so much for a wonderful presentation and taking on these questions. We appreciate you so much.
Thank you very much.
All right, everyone.